Welcome to Have More Babies, the show where we take a deep dive into fascinating topics to make you well-informed quickly. Today we're embarking on what is, well, a truly miraculous journey, the very first step in human development. Our mission is to unpack the incredible story of the zygote, that single foundational cell from which all life begins. We've been exploring a truly insightful article from Omega Pediatrics that lays out this initial stage with such clarity. So let's get right into it. What exactly is a zygote?
It's remarkable, isn't it, how much complexity is packed into this single, often overlooked cellular stage. At its core, the zygote is the genesis of life, a newly formed cell that holds the entire genetic blueprint for a new individual. This deep dive will hopefully offer you a renewed appreciation for the incredible precision required right from the very start.
It truly is mind-boggling. The article from Omega Pediatrics defines a zygote as that initial cell formed the instant two distinct gamete cells, the sperm, and an egg successfully fused during fertilization. It's not just a starting point, it's the singular moment, the absolute genesis containing all the genetic information needed for a brand new, unique individual. Think about it. All your unique traits, from the color of your eyes to your specific predispositions for certain aptitudes or even health conditions, all encoded within that one microscopic cell. It truly is an incredible thought.
Right. And it's more than just a simple coming together of two cells, though. This fusion is a highly precise and intricate combination of 23 chromosomes from the sperm and 23 chromosomes from the egg, resulting in a full set of 23 pairs of chromosomes within that new zygote. This newly combined genetic material doesn't just sit dormant. It immediately activates a cascade of biochemical events, initiating the entire developmental process. It's truly the spark that ignites everything that follows, setting the stage for every cell, tissue, and organ that will eventually form.
And what's fascinating is how the article details the various paths this initial cellular union can take. It highlights two primary ways a zygote can actually form: naturally, through sexual intercourse, or through what we call assisted reproductive technology, or RT. With natural formation, we picture that incredible, almost epic journey a sperm undertakes to meet the egg in the fallopian tube, culminating in that miraculous moment when just one successful sperm penetrates the egg. But science offers incredible assistance too, bridging the gap for so many.
Exactly. Modern medicine has provided alternative avenues to achieve this critical initial cell. Assisted reproductive technology, RT, well, it encompasses a range of procedures. For instance, intrauterine insemination, or IUI, directly assists by placing sperm into the uterus, helping it on its journey towards the egg. Then there's in vitro fertilization, IVF, where that fundamental fertilization step actually happens outside the body in a controlled laboratory setting. Once the egg is fertilized in the lab, forming that zygote, it's then carefully cultured and later implanted back into the uterus. The key takeaway, as the article emphasizes, is that whether it's through natural means or assisted technology, the essential, universal step is always the union of the sperm and the egg.
That union.
That's the critical event that kicks off all subsequent embryo development.
Okay, so once this amazing single cell, this zygote, is formed, how long does this specific foundational stage actually last? The article tells us it's surprisingly brief, typically enduring for only about 4 days.
Just 4 days.
Yeah, but during these fleeting days, this single cell is anything but idle. It undergoes incredibly rapid and continuous cell division, a process called cleavage. It doesn't grow in size initially, but rather subdivides, transforming first into what's called a morula, essentially a solid ball of cells.
A ball of cells.
From there, it quickly reorganizes and develops into a blastocyst, which is a far more differentiated and complex structure.
It's the sheer speed and efficiency of this initial cellular programming that's truly astounding.
Right.
By around day 5 after that initial fertilization, that single zygote has already undergone multiple divisions and reorganizations, developing into a blastocyst. This blastocyst isn't just a simple cluster of cells. It's already a highly organized structure containing distinct cell populations.
Right.
There's an inner cell mass destined to become the embryo itself, and an outer layer, the trophoblast, which will later form the placenta, supporting the entire pregnancy. It's a true marvel of early cellular organization and program development, all unfolding within less than a week.
That distinction between these early stages is so important for understanding the full timeline of human development. The article makes a really clear point about the difference between a zygote and an embryo. To recap, the zygote is that initial single-cell entity right after fertilization, which immediately begins its rapid journey of division.
The single cell.
Exactly. The embryo, on the other hand, technically begins once that blastocyst successfully implants itself into the uterine lining. This crucial implantation marks the official start of a clinically recognized pregnancy.
Okay.
The embryonic stage then continues from implantation until the end of the 8th week of gestation.
Connecting this to the bigger picture, understanding these distinct biological milestones helps us appreciate the immense complexity and progression of early human development. The embryo stage, which follows the zygote and blastocyst, is where all the major organs and fundamental tissues begin to form. This period, from implantation to 8 weeks, is an incredibly intense time of rapid growth, cell migration, and differentiation.
So much happening.
It's when the heart starts beating, the neural tube closes to form the brain and spinal cord, and the basic architecture for every system in the body is laid down. It's the entire foundational blueprint being built out.
Speaking of early development, the article also delves into how twins form, and it's actually quite different depending on whether we're talking about identical or fraternal.
Oh yeah, that's fascinating.
Monozygotic, or identical twins, come from a single fertilized egg, just one zygote. What happens is that this single zygote, usually during the early blastocyst phase, spontaneously splits into two separate bundles of cells.
It just splits.
Right. This division leads to two distinct embryos, both carrying identical genetic makeup and, of course, the same sex. Dizygotic, or fraternal twins, are formed in a completely different way.
Totally different.
They result from two separate eggs being released by the ovaries, and each of those eggs is then fertilized by a different sperm. This process forms two distinct zygotes from the very beginning, each with its own unique genetic code.
What's truly fascinating here is how these fundamental differences at the cellular level explain the profound distinctions between twin types. Identical twins share everything genetically because they arose from that single genetic blueprint, making them, in essence, natural clones.
Exactly.
Fraternal twins, however, are no more alike genetically than any other siblings born at different times. They simply shared a womb. They could even be different sexes, underscoring the power and impact of that initial cellular event, whether it's a single zygote splitting or two entirely separate fertilization events occurring simultaneously. It's a fantastic illustration of the diverse outcomes possible in early human reproduction.
Absolutely. And at the very core of that single zygote, as you said, is its incredible genetic blueprint, the DNA. The article really emphasizes the profound role of those 23 pairs of chromosomes in determining absolutely everything about the developing individual, from observable physical traits like eye and hair color to more subtle characteristics, to genetic predispositions for certain health conditions, and even gender. So when we talk about this, it truly means that little single cell holds the entire detailed instruction manual for a human being. Every single detail, every biological instruction is encoded right there.
It truly is astounding, isn't it, how that complete genetic blueprint is laid down in such a brief moment. The dance of DNA that unfolds from that initial moment is a masterclass in biological programming. Each gene, a segment of that DNA, contains instructions for building proteins, which are the workhorses of the cell.
Right, the building blocks.
The coordinated expression of all those genes guides the transformation of what were once undifferentiated cells into highly specialized structures. Nerve cells, muscle cells, skin cells, eventually forming all the diverse tissues and organs. It's the ultimate example of precision engineering and biochemical programming at a microscopic level, dictating the uniqueness of each individual, right down to the specific enzymes in their cells.
Given how absolutely crucial this early stage is, it's natural to wonder what factors can influence a zygote's development and its ultimate health. The article highlights several key areas, starting, understandably, with maternal health. A mother's nutrition and overall lifestyle choices play an immense role.
Huge role.
It specifically calls out the vital importance of adequate intake of folic acid, which is critical for preventing neural tube defects, and iron, essential for carrying oxygen. Equally imperative is the avoidance of harmful substances like tobacco and alcohol, which are known to have profoundly detrimental effects on a delicate developing zygote and embryo.
You're right, maternal health is paramount, but it's not exclusively about the mother's health. The article also touches on paternal factors, particularly sperm quality, as playing a significant role.
Ah, interesting.
Factors such as a father's age, his lifestyle choices, and even environmental exposures can impact the genetic contribution and the overall health of the sperm, thereby influencing the zygote's formation and subsequent development. Speaking of environmental exposures, the article stresses the inherent vulnerability of the early zygote to external toxins and radiation in its surroundings.
That makes sense.
These microscopic cells, undergoing such rapid division, are incredibly sensitive to disruption. Safeguarding against these environmental hazards is an absolutely vital consideration for expectant parents or those planning a family, as these early stages are so foundational.
Unfortunately, not every journey through these initial stages is smooth, and the article thoughtfully addresses some common concerns that can arise. These include serious issues such as ectopic pregnancies, where implantation happens outside the uterus, most commonly in the fallopian tube, which can be life-threatening if not detected.
Very serious.
It also covers the tragic occurrence of miscarriage, a silent struggle for many families. For anyone who is expecting or planning a family, this information can be incredibly helpful for understanding potential hurdles and complications, and crucially, for knowing when to seek medical advice.
Yeah, absolutely. And it's important context for understanding early development that approximately 20% of pregnancies end before clinical detection.
20%? Wow.
And often, these occur during the incredibly early zygote or embryonic stage, sometimes even before implantation. The primary cause for these early losses, as the article thoroughly explains, is frequently chromosomal abnormalities.
Ah, the genetics again.
Exactly. These are issues like an irregular number of chromosomes, for example, trisomy 18, trisomy 21, which is Down syndrome, or Turner syndrome. In these cases, the genetic blueprint itself has an error, often making further development inviable. It truly underscores the profound fragility and intricate complexity of early life and the astonishing precision required for successful development.
Wow, what an incredible journey we've taken today, from that single tiny zygote to the beginnings of an embryo and all the intricate factors that influence its formation and health. It's a powerful testament to the delicate balance of biology and genetics that creates all of us. We've just scratched the surface of the amazing information presented by Omega Pediatrics in your insightful article.
This exploration really makes you reflect on the sheer wonder of human development, doesn't
It
it?
really does.
It raises an important question for all of us. How can we best support this remarkable process, both individually through healthy choices and collectively through public health initiatives? And what continued scientific discoveries might further illuminate these earliest, most critical moments of life, perhaps offering new insights into preventing complications or supporting successful pregnancies? It's an ongoing journey of learning and wonder, with so much more to explore in this microscopic yet profoundly significant world.
Absolutely. And for even more detailed insights on pediatric care, from these foundational stages of development to comprehensive childhood health topics, you really need to visit omegapediatrics.com. They are an absolutely fantastic resource for parents and parents-to-be, offering expert advice and valuable information that goes well beyond just the zygote.
A great resource.
You'll find everything you need for your children's health journey, from conception awareness right through their growing years. Again, that's omegapediatrics.com, your go-to for trusted pediatric health information. And if you found this deep dive as fascinating as we did, please take a moment to like this video, subscribe to our channel for more insightful episodes, and share it with anyone who's curious about these early stages of life. Thanks for joining us on Have More Babies. We'll catch you next time.